This webinar is the second part in a series covering the blood. This one was going to cover the concept of blood typing. So a person's blood type, blood group, is inherited, and it's determined by the presence of antigens, which are surface proteins, on the red blood cell membrane. The other term for an antigen is agglutinogen, and that term, understanding that term will be important in a little bit. There are a number of antigens on red blood cell membranes, so there are a number of different blood groups, but we're going to talk about the most commonly studied and known, and we're talking about the ABO and the RH blood groups. So we'll talk about the A and B antigens in regards to ABO blood typing, and the RH or the D antigen when we're talking about the RH blood typing. Some antigens will cause formation of antibodies that are opposite to the blood type that the person has, and those antibodies circulate in the plasma, and they're referred to as agglutinins. So for example, if someone, say, is a blood type A, they would have antibodies against the B antigen. So they have antibodies that are against things that they don't have. The first question, though, is what's the significance of blood groups, blood types? Well, apparently there isn't any apparent significance physiologically. Where it becomes important is when someone needs a blood transfusion. So understanding blood typing is important when a person needs a blood transfusion. So first is, why should we transfuse blood? Well, transfusion of blood is usually used to replenish the oxygen carrying capacity of the blood. So if someone has anemia or blood loss due to surgery or trauma, they're going to transfuse them with blood. So it's going to be important that they get the right blood type. So what is transfused? Well, a blood transfusion is actually a little bit of a misnomer. When a person has a blood transfusion, what is actually given to them is washed, preserved red blood cells. Plasma is not administered. They're giving the washed, preserved red blood cells. The reason for not giving them or minimizing the amount of plasma, so it'll minimize any potential problems in regards to blood mismatch. So the typing, why do we type the blood? Because of what potentially can happen if you give someone the wrong type of blood. So if you see the picture on the bottom, what can happen is if someone, and I'm gonna label this, so this would represent who's going to be receiving blood. So if a recipient has antibodies against, say, the B antigen in their blood, and they're given blood cells from someone who has the B antigen on the red blood cell surface, what can happen is a situation referred to as agglutination. That's why I wanted you to remember that word, agglutinins. So agglutination is where the red blood cells will clump together, and what happens is when they clump together, they're gonna weaken. They can hemolyze, and as they hemolyze, they're going to block capillaries and impair a person's ability to get blood flow to certain organs, and also the hemolysis of the blood cells will lead to a lack of oxygen carrying capacity in their blood. So what we're trying to avoid when someone's getting transfused with blood is a transfusion reaction. Now that can be, in what you see here, because of the hemolysis and the clumping, it could be a fatal condition. So we're going to always try to properly match the blood types. So let's kind of start with kind of a little bit of genetics first before we go over the various blood types. Well, actually, we're going to start with genetics with the ABO blood type. Now, I had mentioned earlier that the ABO blood type, or all blood types, are inherited. The ABO blood type is determined by specific antigens on the person's red blood cells. So with the ABO blood type, every person will inherit two alleles, or I would say two versions of a gene from a parent. So they're going to inherit one version from, say, the father and another version from the mother. Well, there's three variants or three alleles for the ABO blood typing. There's the A, the B, and the O. Now, the A and B alleles are considered to be codominant. So if this individual, which you see right here, has inherited an A allele from his dad and the B allele from his mom, both will be expressed. So this person would be blood type AB. An individual with type O, which you see over here, has inherited an O allele from both mom and dad, and they're considered to be homozygous for the O allele. Now the O is recessive to A and B. So in order for someone to be type O, they have to contain both alleles, the O alleles. And these two individuals you see here, they inherited a O allele from a parent and another allele from their mom. So the one to the first left inherited the A from the dad, and the third child inherited the B allele from the mom. So because O is recessive to either A or B, if you have an A or B allele, you will have B type A and B respectively. So let's kind of look at it again just here so you can see in the chart that you have. So I want to distinguish between a phenotype and a genotype. A genotype is your genetic makeup. So these are the variants, the alleles they inherited from their parents. The phenotype is what they outwardly express. So being type blood O, this here, the little I's represent they inherited O alleles from both parents. So they are homozygous for the O allele, they're O, type O blood. Type AB, they have both an A and a B allele, and since it's codominantly expressed, they are type A and B blood. If I am type A, I have two possibilities for genotypes. I could be homozygous for the A allele, they've received A alleles from both parents, or they're heterozygous. They received an A allele from one parent and the O allele from the other. And since O is recessive to either A or B, this person would be type A blood or A blood. And then if you look at someone who's a type B blood, they could either be homozygous for the B allele or heterozygous. Okay, so there's possibilities here. So if we kind of go to the next picture, let's kind of emphasize or to reaffirm what the ABO blood typing means. So the blood types are determined by the presence of or absence of antigens on the red blood cell membrane. So this person to the far left, you see the red blood cell. The little red circles, we're going to say that's the A antigen. So a person with type A blood type has the A antigen on the red blood cell membrane. Actually, let's erase that so we're going to be consistent with the colors. So this is type A. The second person is type B. And these little blue triangles on the red blood cell membrane is trying to represent antigen B. So someone with type B blood has the B antigen expressed on the red blood cell membrane. A person with AB has both antigens expressed, and a person with type O doesn't have either A or B antigen expressed on the red blood cell membrane. Remember that a person with a certain blood type or certain antigens on the red blood cell membrane will develop antibodies against antigens that they normally don't express on the red blood cell membrane. And in regards to the ABO blood typing, this development of antibodies occurs shortly after birth. We don't understand the reason for it. Some believe it's something to do with what you eat and bacteria in your GI tract somehow is going to result in your body's production of these antibodies, but no one really knows for sure why they develop. But a person with type A blood will spontaneously develop antibodies against B. So these blue triangles, or not triangles, or Ys, represent anti-B antibodies. So they're going to be floating around in the blood plasma of this individual. People with blood type B have anti-A antibodies in their plasma. People with AB, they don't have either because they express both A and B. And the last individual who's type O, since they don't express either A or B on the red blood cell membrane, they'll have both anti-A, which you see here, and anti-B antibodies in their plasma. The most common blood type in the world is type O. If you look at just the pie charts, you can just visually see that majority is type O. So most common blood type is type O. The least common blood type is AB. But you'll notice that there's variations in the distribution of blood types depending upon either your racial or ethnic background. So if you notice with Native Americans and the Aborigines from Australia, they have a preponderance of type O. And like the Native Americans, it's either type O or they're type A. And the Aborigines, it's a large percent, probably over 45% are type O, and then you have a little bit of A and B. So let's go to another blood type, is the Rh blood group. The Rh blood group is determined by the presence or absence of the D antigen on a red blood cell membrane. Now, the reason why they call it the Rh factor, because it was originally studied in rhesus monkeys, and rhesus is spelled R-H-E-S-U-S. So the Rh factor is, if you're Rh positive versus Rh negative, is determined by whether or not you have that D antigen. So a person who is Rh positive, which you see right here, this person would be considered Rh positive. They express the D antigen on the red blood cell membrane. If you're Rh negative, you do not have the D antigen. Now, the genetics of the Rh blood group is that you have either, if you're Rh positive, you have both D alleles, or which is mean you'd be homozygous for the D allele, or you're heterozygous. You have the D antigen or you don't. This is going to be a simple dominant recessive relationship. There's no codominance here. So in order to be Rh negative, you have to be homozygous for we're going to put the little d. So if I have little d, that means I inherited alleles from both mom and dad, and neither of those alleles will be able to express a D antigen. So if you're Rh positive, you have two possible genotypes that will make you phenotypically Rh positive. If you're Rh negative, you can only be homozygous for the little d allele. Now with the Rh factor, it's a little different from the ABO blood group, is antibodies do not form spontaneously after birth. I am personally Rh negative, and I do not have antibodies against the D antigen, but I can if you either transfuse me with blood that has the D antigen on it. I will start to develop antibodies against it. Or if I we'll show you an example of what also could result in the production of antibodies, is if I'm carrying an Rh positive fetus, there is a possibility that I can develop antibodies. So in other words, for someone who is Rh negative to develop antibodies against the D antigen, they have to be exposed to Rh positive blood. So I'm going to show you something that could happen. Is an Rh negative mother marries an Rh positive father. They have a child. She becomes pregnant. If the father's Rh positive, there's a 50 to 100% chance that she will carry an Rh positive fetus, because is if he is Rh positive, he is this or he is this. Remember, the mother is this for her genotype. So there is actually, if he is homozygous, it's a 100% chance she'll carry an Rh positive fetus. If he's heterozygous, there is a 50% chance that she will carry a Rh positive fetus. So during pregnancy, what can happen is if there is some sort of trauma or mixing of the babies in the mother's blood, which is usually, which they have in here, is usually often it'll happen at delivery because that's going to be the most traumatic. But anytime if there's a mixing of the child's and mother's blood, she will develop antibodies against the D antigen that will be able to identify and bind to the D antigen on red blood cell membranes. And it takes some time. So the first birth will be uneventful. So the development of those antibodies can take some time. So the first child is safe. But the thing is, if in a later pregnancy she carries an Rh positive fetus, she's already had the antibodies and she has cells that recognize that these D antigens, and they can start to ramp up and produce a whole bunch more antibodies that will ultimately go and attack the baby's red blood cells, and what would result is what we refer to as hemolytic disease of the newborn. Now, thing is, and this kind of goes through the same thing, but it illustrates a couple things that I want to talk about here. So what the hemolytic disease of the newborn is not really a significant problem nowadays, just because the advent of medicine, that with there's a drug called Rogam, which you see here, that is administered to a Rh negative woman who is carrying a baby. It's usually administered about the 28th week of pregnancy, and then it'll also, another one will be administered within about 72 hours after childbirth, specifically if the child is found to be Rh positive. What happens is the Rogam is an antibody. It's actually an IgG antibody that will take out any Rh positive red blood cells that may have been introduced into the maternal bloodstream from the baby before the mother has a chance to react to them and develop the antibody. So what they're trying to do is prevent the mother from developing antibodies against the D antigen. The question, though, comes down to is if this Rh factor incompatibility is a problem, why isn't there a problem between ABO? So you could be type A and your child could be type B. We never seem to talk about that, but we always talk about this Rh factor. Well, the reason is that the AB antibodies are fairly large, and they are of an IgM type antibody. And the IgM type antibodies do not cross the placenta. So it's rarely an issue. The ones for the D antigen are IgG, which do cross the placenta. So this is my first introduction to the different types of antibodies, because we'll be discussing this with the lymphatic and immune system. So the reason why ABO blood typing is not much of an issue or mismatch between mother and fetus is because it's the antibodies are IgM, they don't cross the placenta, but IgG can. So if a mother is carrying an Rh positive fetus, the antibodies that she has can cross and attack the child's red blood cells. So what I want to do next is talk about blood typing, and then what happens if the significance of blood typing again, and what would happen if it's not properly matched. So blood typing, and what you're going to do in lab is you're going to have these little plates, and they're going to be labeled, these little wells. There's an A, there's a B, and the other one they'll label as RH. What you're going to do is you're going to put a few drops of this anti-serum for either A anti-serum is going to go in this well, anti-B anti-serum is going to go in this well, and the anti-D anti-serum is going to go in that well. So we're already going to put in technically antibodies into each well because we don't know the subject's blood type. And remember, a subject's blood type is determined by the presence or absence of antigens on the red blood cell membrane. So you have your respective antibodies in each well. You're going to take a sample of blood and you're going to put a drop of that blood into each respective well. You will mix them, and as they mix, if the antigen that is, or I should say, let's put it this way, if you have a particular antibody in that well, if the antigen for that antibody is found on the red blood cell membrane of the subject, they will clump. You will have an agglutination reaction, just like I had mentioned earlier. So you will be able to figure out a subject's blood type based on what you see in the well. So if you look here, this is actually a real one. You have a blood sample, and there's your blood. In these wells, they put the anti-A antibody. In these wells, they put the anti-B. In this, they put the anti-D, or the antibody for the Rh factor. They mix the samples, and what you'll see here and here, all this, these are agglutination reactions. So that meant let's look at this row, that this person is A positive. That means they have the A antigen and they have the D antigen on the red blood cell membrane. That's why we saw this clumping reaction, or this agglutination reaction, in those respective wells. You'll see someone who's B positive. You see the clumping here. AB positive, you saw it in all three wells. If someone is, say, O negative, that you're not going to see any type of agglutination reaction in any of the wells. So using the idea or the knowledge of, I know the person's blood type is based on the presence or absence of a particular antigen. If you put a known antibody in a well, mix it with a sample of blood, if there is clumping, you know that antigen is present. So what I'm going to have you do is you're going to fill in this chart by yourself. And so you know how to fill in the chart is this row right here is going to represent a recipient. Say I'm in surgery and I'm A negative because I am A negative. I want to know which blood types I can be given if I need some blood. So these are the recipients. These are the possible donors. I do want you to understand, though, under any condition, they will try to perfectly match you with your blood type. So if I'm A negative, they're going to give me A negative blood. But I want you to look at the other choices and say, who possibly under an emergency situation I can also be given blood from. But also, it's very important that you realize if someone is A, or if it's Rh negative, they will not give them Rh positive blood under any circumstances. Because I'll get people that say, well, remember the production of the antibodies takes some time, so that first time it's not going to matter. You don't know if that's happened to the first time for them. You don't know that I have antibodies against the D antigen. So you will never give an individual who is Rh negative blood Rh positive. So don't even go there, okay? So I want you to do is fill in the chart, put an X in the boxes, and I'll at least do this one for you. Say I'm going to only do one of it. That A negative blood can get A negative. So you put an X where I could get blood from a particular individual. So remember, when you're trying to figure out transfusions, is always consider the antibodies present in the recipient's plasma. I have the antibodies against the B antigen. So you do not want to give me blood that has the B antigen on it. Also remember, since I'm A negative, I don't have antibodies against the D antigen, but you don't want me to start developing it. So you're never going to give me Rh positive blood. So remember, it's always think of the antibodies that are present in the recipient's blood when you figure out whose blood type you're going to be administered, because they're going to give me the preserved washed red blood cells. They're not going to give me the plasma, so I don't have to worry about the donor's antibodies. I just have to worry about the donor's antigens. So use what I just told you to be able to fill in the chart. And then I want you to determine which individual on this chart, which of the specific blood type, A positive, A negative, B positive, B negative, blah, blah, blah. Who is the universal donor? Who can be given blood to anybody? These are the people they call and hound and say, can you donate blood? Can you donate blood? Can you donate blood? Because they are the universal donor. My niece is the universal donor, so she's always being asked to donate blood. But what the thing is, what screws her is the fact that she can only be given blood by her particular blood type. But thankfully, it's a little more common blood type. But so you'll see that when you work out the chart. Then also determine who's the universal recipient. Which blood type can get blood from everybody? So if they're in need of blood, they can get it from anybody because they have no problems receiving blood from any other individual. So I want you to fill that out. So actually, I do want to go back to this picture just again to emphasize, if I can find it, that the idea of the mismatching of blood. So this first scenario, which you see here, was a person with type B blood. They have the B antigen on the red blood cell membrane. But I had mentioned here, this is the recipient. Actually, no, they're type A, sorry. This would be me. This is type A blood. I have these anti-B antibodies in my bloodstream. If I receive this blood from someone who is type B, the antibody will find the antigen. You get this clumping, this agglutination, which weakens the blood cells. They hemolyze, and as they hemolyze, they're going to block or damage certain blood vessels in the body, and it could cause significant damage in tissues. And one thing I do want to, you can write this at the very end of your notes, is what also happens is these antigen antibody complexes, as they form, they're going to activate a plasma proteins called complement proteins, which will be also involved in this whole process of the hemolysis of the blood cells. The reason why I'm bringing this up, because we'll discuss this when we do the immune system. So this is the end of the second part of the webinar series.